speaker series. I am Len Friedman. I'm the Chief Science Officer here at the Frederick National Laboratory for Cancer Research. And it's really, again, a pleasure to be here and to have you all here this morning. The Biotech Connector event is a collaboration between the Frederick National Lab for Cancer Research and the Frederick County Chamber of Commerce. It's a great event that promotes and supports Frederick County and surrounding areas, biotech and bioscience community, and it highlights recent local breakthroughs and brings an opportunity to network here with other scientists and entrepreneurs. With Biotech Connector spotlighting local biotech progress and network, I'd like to just take a second and make a shameless plug for an event we are planning in Frederick for June of 2026. And that is, we here at Frederick National Lab have co-sponsored three previous symposia on different topics that relate to cancer research at Hood College. And the next event, as I said, it's June 16th through June 18th, is a symposium that's going to be focused specifically on reproducibility in research, in biomedical research. And this is an extremely relevant, important, and timely topic. This meeting will cover a limited number of specific areas. They include research antibodies, protein reagents, drug discovery, organoids, AI, and a few specific aspects of human genetics. But I really encourage everyone here in the room and online, when we open up registration, and that won't be until, I think, January, to consider attending this, as I said, extremely important event. And I should also say that the focus of this particular meeting, as it relates to reproducibility, which is obviously a huge, huge topic, is how specifically standards and reference materials in these specific areas can really, I think, make a positive, have a positive influence on increasing reproducibility. So keep your eyes open for that event in mid-June of 2026. Whoops, that was not what I wanted to do. So I'd like to now introduce our three distinguished speakers for this morning, who will dive into recent developments in cell and gene therapy, which is the focus of this morning's Biotech Connector. Vinay Vyas is the Associate Director at the Biopharmaceutical Development Program, BDP we call it, here at the Frederick National Lab, where he heads the development group in the process development and clinical manufacture of ADCs, vaccines, cytokines, viral vectors, and cell and gene therapy products. More recently, he spearheaded the establishment of BDP's cGMP CAR T-cell therapy facility, enabling the Frederick National Lab to rapidly become a leading manufacturing site for the NCI's clinical trials. Vinay graduated with bachelor's and master's degrees in chemical engineering and chemistry, followed by a Ph.D. in biotechnology and biochemical engineering. He has over 30 years of progressive experience in bioprocess development and translational research. Our second speaker is Jon Rowley. He is the founder and chief product officer of RoosterBio and has a goal of making significant contributions to the commercial translation of living cellular technologies. Jon received his Ph.D. from the University of Michigan, go blue, in biomedical engineering, and has authored over 40 peer-reviewed manuscripts and 20 issued or pending patents related to biomaterials development, tissue engineering, and cell therapy manufacturing. Jon is also on the board of directors of FITCI, Frederick's local technology incubator. Our third and final speaker is Brian Paszkiet. He is the principal clinical scientist at Miltenyi Biotech. Brian is an experienced scientist specializing in cellular processing, gene therapy, and automation. He holds a master's degree in microbiology from the University of Illinois and over 2 decades of experience. And he has held pivotal roles in Miltenyi Biotech, Thermo Fisher, and other leading organizations. At Miltenyi, Brian excels in creating and testing automated processes for the CliniMACS Prodigy and supports customer service across the United States in automating their complex processes. Brian has contributed to numerous publications, patents, and product launches in the cell therapy and gene therapy field. So with that, I would again like to welcome everyone here in person and online this morning, and I'd like to welcome our first speaker, Dr. Vinay Vyas.
Thank you, all of you, for inviting me here. Oh, sorry. Yeah, thank you for the opportunity to speak. I've been giving this talk a couple of times. The last time I probably gave was probably in 2018-ish, when the earlier parts of the Biotech Connector. So this is the second one, and also on cell and gene therapy. This one works.
Go one more.
Just go. Okay. So BDP has been in the forefront of cell and gene therapy since we established it in 2018-ish. We got the opportunity to do this when NCI tasked us to set up a facility. And the Biopharmaceutical Development Program, for some of you who are not familiar with it, is part of the FNLCR. We are a unique entity within the FNLCR. We work like a boutique biopharmaceutical company. We have our own QC, we have our own QA, we have a manufacturing unit and a process development unit. We are funded entirely by the National Cancer Institute, and we are overseen by the Biological Resources branch, which is part of DTP and DCTD under the NCI. We are all Leidos employees. We operate under a GOCO, government-owned and contractor-operated. And our mission is to develop novel therapeutics for treatment of cancer and other diseases. So this looks slightly top-heavy towards process development because I'm part of process development, but we have a very strong manufacturing, QC, and QARA teams. As part of process development, we do all kinds of process development, completely platform agnostic. Apart from cell and gene therapy, we do ADCs, we do monoclonal antibodies, viral vectors, and other bacterial cell culture, you name it, we do all the processes. Coming to cell and gene therapy, we have a very strong program in CAR-T and TCR-Ts. And I'll just go through a quick list of CAR-Ts and TCR-Ts that we are currently working on. We started off with a CD33, where we got our feet wet using the CD33. This initial vector was not made in BDP. We got it from CHOP. But we used that, we leveraged that information and developed our own platforms. So that was starting with CD33 in 2018-ish. And then we got a lot of projects from the NCI for IL-2, mesothelin CAR-T, which is currently ongoing at the clinical center at NIH. STEAP1, another unique one where they wanted us to do a 1:1 CD4 and CD8. Against a metastatic castration-resistant prostate cancer. And currently we are working on mesothelin FAP CAR team, which is also a T-cell engaging secretory molecule. And we have an interesting project that does a TCR-1188, which is part of the KRAS mutated solid tumors. These are all lentiviral-based CARs or TCRs. And we also have a very big list of retroviral-based CARs and TCRs. The CD123 is a bispecific T-cell engaging molecule that is secreted out against CD123 myeloid malignancies. We have another one, TCR, CD22 TCR, which was part of the NCI, Dr. Ishii Kazuza. I believe she's not there anymore, but I guess they're going to do the clinical trials shortly. So So as I said, we have a number of CARs and TCRs in the pipeline, past and present. So this is just a quick, a unique CAR that the process that we developed, which essentially wanted an equal ratio of CD4s and CD8s. So to achieve that, we had to isolate CD4s and CD8s separately, process them parallelly, and move from the Prodigy to G-Rex because the deliverables were a very large amount. And finally, when we harvested, we again had to formulate it so that we finally have a 1:1 CAR at the end. So it was a pretty complex process, and our manufacturing team managed to accomplish that very well. And we've got a few, it's currently ongoing, and I think we have done 3 or 4 patient runs already. This is another TCR. It's a UPenn, a PI from UPenn. So this involves not only lentiviral transduction, we also have 3 knockouts. So after a lentiviral transduction, it gets base edited on a electroporator using guide RNAs, and then goes into cell expansion followed by harvesting. And we also have an in-house process for CRISPR-based knock-in and knockout, which essentially uses a electroporator again to transfer the gene of interest as well as to knockout. So we have 3 different processes that kinds of processes that anyone could leverage. All this needs the engine of CAR-T is typically viral vector, and we have developed in-house viral vectors, lenti and retroviral vectors, processes for the same. And this took a while because we had to start from scratch. We had to develop we had to identify, source the plasmids. We had to identify, source the cell lines. We had to make GMP plasmids, or at least source GMP plasmids. We had to make the GMP master cell banks. So all this took quite some time, and then we had to develop the process. So we had both lenti and retroviral based vectors in place to help with the CAR-T process. This slide just shows you the testing that we have to do to release the lentiviral vectors, a typical identity, potency, purity, and safety, which most of you are probably familiar with. And we have a QC team that ensures that we have a qualified assay for all of these tests. And similarly, for a cell product, we have a spec. We define a spec based on the incoming project, and then we test it and release it finally, QA releases it. I just wanted to briefly put a slide on AAV9s that we have worked on. And we have a process. So typically, the AAVs are purified using an ultracentrifuge, which is the gold standard for purification to separate out the empty versus full capsids. But we have developed a chromatographic process that is more easily scalable compared to the centrifugal process, which is limited to how much you can centrifuge and purify. So we have a good platform, and we have scaled this up to a 10-liter scale, and it works pretty well. So I thought I'll quickly show some of our latest facilities. This is one of our manufacturing suites. It's area B3. You can see we have a wave bioreactor, and in the room, there's a close-up of that. There's a Prodigy. There's a plug for Miltenyi. Better pay me for that. So, and here we have a virus suite where you can see a disposable bioreactor inside the suite, and all the hookups. So we can do a viral vector or a AAV project inside. This is just another picture to show the Prodigy inside the room. This is definitely a plug for you. Yeah, and some more pictures. And you can access our program through the next program, NCI's next program, which is next.cancer.gov. Questions?
I got a question.
Yeah.
Great stuff. Thank you so much. I love seeing all the cell therapy stuff going on in right here in Frederick. And Jon Rowley from RoosterBio. So I'm a process development guy too. As you get the T cell process that you were working on, that was being base edited, right?
Yeah.
I mean, that must come with kind of new process challenges, and then also you said you were using a electroporator. Are there special outside of Max that maybe you're using Maxite, but are there other like
So there are others.
clinical?
Yeah, there are
electroporators
others.
out there?
I mean, Biorad makes them. I mean, there's different brands.
Okay.
You could use any of them. Nuclear factors. Miltenyi is probably working on one too. It's an electroporator. So
Yeah. And what was the biggest challenge in the base editing? Is it just C? Is it in the like does it add a lot of time to the process?
So base editing, you need to optimize base editing. So majority of your product is base edited, right? So because you cannot separate out the non-base edited from the base edited. So you need to optimize the process to get efficient base editing. So we get close to 95+% base edited.
Wow. That's awesome. Okay.
Yeah.
There's somebody online.
Yeah. Hi, Vinay. It is a great presentation. So my question would be for the retroviral and lentiviral vector programs, what is the current strategy for accessing the risk of insertional mutagenesis? And what are the methods employed to reduce the risk, in the clinical products ultimately?
So that's actually a challenge with all viral vectors. So
Right.
there is going to be some of that. So unless you do sequencing, that's why the FDA kind of asks you to monitor the patient for n number of years to look out for any of these mutations. And that is definitely one of those pitfalls of using viral vectors, where they could be integrating in regions that are not the ideal regions.
Okay. So is this like the sequencing thing, or looking for the mutations after when it is delivered to the patient? Is it included in the process, or is it something that that the doctors would ask for later? Or
Yeah, currently it is not included. You could probably do a whole genome sequencing of the product if it gets fast enough. But currently, since we have a turnaround of about 14 days from the arm to arm, or close to 3 weeks from arm to arm, it's very difficult to do most of these testing, sequencing, and turn it around within that short period of time.
All right. Thank you. Thank you so much.
Thank you.
I think I'm next. I'm Jon Rowley, RoosterBio. Just
To use the laser pointer, it's right there.
okay. All right. I talk loud, so if I need that closer, let me know. All right. So I'm Jon Rowley. I started up RoosterBio back in 2013, and we're here mostly to industrialize the supply chain for cell and gene therapy. And we started with mesenchymal stromal cells, which is definitely one of the most widely translated cells into the clinic. We started RoosterBio, really to reimagine a highly fragmented supply chain in cell and gene therapy, right? And I got to know that because, for the 10 years before I started RoosterBio, I worked at therapeutic companies and then spent 5 years here at Lonza, helping tens of companies get into the clinic with a manufacturing process. And I'm a process development guy as well, right? So I was just telling Mark that it's definitely one of the more fun jobs as an engineer in cell and gene therapy. And so what we try to do is we take a process first approach towards product development, and we've now started a products company. And so what we've done when I say we take this process first approach is we designed our own scalable manufacturing process, built for flexibility and various stages of clinical development for human mesenchymal stromal cells. And then we own all the pain in the butt stuff that really holds therapeutic companies back and adds years and years of time to their development, which again, I've learned by being at therapeutic companies and then at Lonza. We manage all of the tissue isolation at the front, tissue acquisition. There's a lot of regulatory red tape around tissue acquisition, isolating the cells, creating master cell banks, creating working cell banks, and then this scalable manufacturing process for the bulk MSCs that can go into any therapeutic program, and then we productize this, right? So our products are these high volume MSC banks. And what those are working cell banks within the context of a manufacturing process. There's a lot of companies that sell bottles of cells and vials of cells and bottles of media, but those are all for basic research, right? So these are specifically for therapeutic product development. We have this high efficiency, high productivity bioprocess media for expanding the MSCs into a final product. And we have these now all in, there's GMP formats in almost all of our products that we sell under the CliniControl brand name. And then as we were, establishing these base processes and products to execute them, and then, essentially we give these manufacturing processes away for free to our customers. That's kind of like the razor of the razor blade business model. And then, they can execute those if they use our cells and our media, right? So that's like the business model that we've established. And then MSCs are used extensively for exosome or extracellular vesicle production, and so we've established specialty media in EV for EV production, as well as for genetic engineering. And again, we design those products to fit within the context of a therapeutic manufacturing process, right? And we have dozens and dozens of posters on our website that we present at national meetings. And so if you're interested in any of those, I'm sure you can get a lot of information from our blog or from our posters. And then we have a process development services group that's focused on our products that helps our customers establish GMP processes for the products that they want to take into the clinic, based off of our products. And then what our customers do is they overlay their intellectual property on top of our base process, right? Whether it's genetic engineering those cells for a cell-based gene therapy, whether it's expanding the cells and MSCs are used in tissue engineering, and they bioprint a piece of cartilage or bone. They will, make those cells and collect the exosomes. Often engineering the exosomes either by engineering the cell or engineering the exosome post-production. And then we help them establish that scalable process, tech transfer to one of our partners, CDMO final product manufacturing. We're about 50 people. I think we're over 50 people now. We had some growth this year, and really this mission of fueling the rapid commercialization of scalable, regenerative medicines is what we're trying to do right here in Frederick. So MSCs, they're really a workhorse of regenerative medicine. If you're not familiar with them, they're for foundational technology for several different approaches. It's because they do a lot of things. Everyone's got them in their bodies, and they are general tissue repair cells. You got a problem, it gets stimulated, it goes to that site, and it helps you fix it. There's several regenerative medicine products that are being used. They're also extensively used in immune modulation, and this is where they first got into the clinic in the early 2000s. using it to damp down the immune response to graft versus host disease. But they're really used in a variety of applications here, from traumatic brain injury to all sorts of things, where really modulating that immune response to trauma is really, really important. They are foundational cells for many of the musculoskeletal tissues, things like bone, cartilage, tendon, and ligament. So you'll find these as raw materials in those areas. Again, people are modifying these with genes for cell-based gene therapy. And then these MSC-derived materials are the exosomes or the secretome that these cells secrete for, people are kind of selling as cell-free therapies. They're easier to keep alive, and, well, you don't have to keep them alive. It's just a better, cold chain that is used. And then, we have really built up a large business in the extracellular vesicle production. And EVs, I won't really get into them that much, but they're secreted by the cells, and they have many of the therapeutic properties. And there's several publications where these extracellular vesicles that are secreted by EVs, even before engineering, are used in really all of the different indications that MSCs have been found in. And MSCs finally got approval in the United States. There's probably a dozen products worldwide that are clinically approved, and Mesoblast finally got their product approved earlier this year. And so the biggest challenge that we're really addressing here, and something that I lived myself again for 10 years before starting RoosterBio, is that developing advanced therapy products really takes several years. And one thing I learned is that if you're a therapeutic company and you already have your base manufacturing process and base cell banks already down, you can start a clinical trial like that, right? It's really just establishing the base preclinical models, and you can get into GMP manufacturing very, very quickly. But if you're not, you are 5-10 years away from getting into the clinic, and this is what we shortcut by having these off-the-shelf cell banks that you can and then the products that you can rapidly do product development and rapidly get into clinical development. And because it takes so long, this really been holding the entire industry back. And now we've helped dozens of companies, get through this in a matter of a couple of years. So, what we've found, and really the business hypothesis when we started, and that we've proven, is that by having standardized MSC production platforms and cell banks, it could really radically simplify therapeutic product development. First, by making MSCs, anybody that wants to take a therapeutic approach and bring MSCs into their lab, they can very quickly get up and running because it already has this production process made. Once you know what you want to do, then you want to get into product and process development, which is where the engineers come in and really scale it up and put the product specifications around it. Having cell banks that are similar to your clinical cell bank is really, really needed. And then having the off-the-shelf processes is also super critical, because now you can spend all your time on product development and less time on process development, and those two are really intertwined. And so if you already have that process, product development is where you can focus your time, which is really the value add for the therapeutic companies. And then once you're ready to get into the clinic, having off-the-shelf cell banks really saves you years of time, because it can take a very long time to put down a good cell bank. And then really the manufacturing platform drives then the final cost. And if you are have ever looked into cell and gene therapy, all of the investment field, they say cell and gene therapy has three main problems: manufacturing, and manufacturing, right? And I can't tell you how many times I've seen that at JP Morgan, right? But it really is that these cell gene therapies, they're expensive. They're expensive because they take a very long time to develop, and then once you actually have it developed, the cells are horribly expensive. But as you scale, you can bring these costs down, right? It's kind of like the Moore's law applied to cell therapy, right? And as you what you have here is the lot size, different manufacturing platforms at the bottom, the lot size in a log scale. And these technology S-curves are very common in the innovation literature, but it applies across all technologies, applies to cells. And we've been driving this and getting customers into the tens of billions of lot sizes, again, using our base processes, and then really managing these costs as the therapy progresses through the clinic, and then we have several blogs that outline really how you can go through this for your specific product. And then again, the challenge that we help is that it takes several $1 million and years of time to take the process development programs through these, and we've done this over the last 10 years with venture capital funding as well as some government contracts and good collaborators. And so we're able to help people get into a variety of yields depending on where they are in R&D, product development, or clinical development, and both with cells and EVs, which are kind of just considered particles that you produce. And then, just to wrap up, we did a collaboration with Sartorius several years ago. We got a lot of posters on our website about this, right? But we really worked on establishing these scalable processes in their off-the-shelf bioreactors, which are used in monoclonal antibody production and lots of different bioprocesses. We went and optimized in their AMBER 250, went through all the different critical process parameters, and then scaled it up into their 50-liter SDR bioreactor. And really got good comparability between the small scale and the large scale, both in bone marrow and umbilical cord. Sorry, I thought that second one was umbilical cord. But then also went through what's really critical is that the cells, after you scale them up, maintain all of their biological properties, right? We didn't have a therapeutic indication in mind, but the general properties that we focus on are just the surface markers. This is the difference between the bioreactor cells or the 2D control. Maintain their biomarkers. This IDO is an immunomodulatory measure of MSCs that they are maintained. They're also used in a lot of angiogenic type indications, and so we follow a host of angiogenic cytokines. The traditional osteogenic, adipogenic, and chondrogenic indications are less important for most of the stuff going on in the clinic today, unless you're in tissue engineering, but these are also maintained. And so, just to wrap up, we have these base manufacturing processes at a variety of scales that, again, we've helped dozens of companies get into clinical development and hoping to go commercial. And we're able to help people go through this very lengthy development program to get to their first-in-man testing and really achieve this in just a couple of years. So I will, with that, I'll take questions, and you can go to our website. We have lots of educational information out there. So thanks. Yes.
Any sort of standards that exist around production? Are there areas? It sounds like you have a great handle on that, see, but is this something that's needed in the field for production?
Standards are very important, right? And I mean, in every piece of this, right? But I mean, there's a lot of standards around just even tissue acquisition and cell banking, right? I try to. I'm more of an innovation guy, right? I like to focus on kind of the scale up and everything, but I mean, there's very clear guidelines that the FDA just has. Now these are guidelines, right? There's no rules, right? But as long as you're responsible when you're doing your scale up, right, and you're really looking at the science, and when you scale up, I think what's really important to understand is that comparability of your product from a 6-well plate or a T75 out of a bioreactor, they're going to be different products, right? There's a lot of differences, but are the critical quality attributes that relate to the therapeutic potency and efficacy and safety all maintained? And that's really, I think, the key thing to think about. The standards is such a broad field. I don't know how I can really answer it, but yes, there's standards all across therapeutic development that need to be maintained. And as long as you do it responsibly, as you scale up your product and understanding what the potency attributes are really hitting your indication, right? Is to me, the most important thing. Anything online? How do you?
Got a question. Carter from Camp.
Hi, Jon. Hi again. A great presentation, and my question would be a little technical, but I
Sure.
would still take my chance. So, I read about the cyclic GMP media and raw materials that RoosterBio is producing, or so. Is there any modification to cyclic GMP, because that is a activator of a STING pathway, and so I just want to understand.
That's a great question. CGMP stands for not cyclic GMP, which is a biochemical term. It's current good manufacturing practices.
Ah, okay.
I actually, Mohaderen just I saw a LinkedIn post from him arguing that the C needs to be capitalized, and maybe I should do that so it doesn't look like it's cyclic GMP. But yeah, it's actually current good manufacturing practices, which are part of the Code of Federal Regulations. So sorry for that confusion.
No, and I mean, thank you for the explanation.
Sure. Carter.
Jon, so I'm curious about the downstream process for these. How do you handle from the small scale all the way to the scale bioreactor, I guess, harvesting the cells and ensuring that you don't have any introduction of adventitious agents? So I assume.
Yeah, you could do it all closed system. There's closed system. There's a lot of counterflow centrifugation or large scale centrifugation type devices that are on the market. But for the cells, we grow the cells on microcarriers. So you add triple usually, and to separate the cells from microcarriers, pass them through a large screen, which brings the microcarriers out in any clumps of cells. And then there's things like the Rotea that Thermo has, which is on a smaller scale. There's the KSEP, which is more scalable, that can do tens of liters to hundreds of liters, that can then remove the cell from the triple and then get a new reagent in there that the cells can then be formulated for cryopreservation.
So then post-carrier removal clumpage is a major concern, and then is that something that's empirically determined based off of your processes, or have you already identified that using these cells that you guys are selling?
Yeah, we've done several 50-liter harvests, and you just have to manage the the clumping of the cells, but you can remove them with 100 micron filters, right? So yes.
So outside of the government, medical centers, most cell-based programs have another hurdle, which is it's a non-reimbursable. And so what I was curious about is, can you give to the clinical trialists a ready-made cell-based product that can be infused into patients, or do they have to then take your product and then modify it? Because when I was provost at MD Anderson, we would spend huge amounts of funds actually producing cell-based products that was non-reimbursable. And the equation was, once you infuse it in patients, that becomes reimbursable, but it becomes a barrier that most medical centers just can't afford.
Yeah, it's so no, we can't. And the reason for that.
You see the barrier that I'm talking about. That's really why.
Yeah, I can't solve.
It can't be democratized unless you can solve that problem.
Yeah, there's a lot of problems to solve. I think that one is possible to be addressed, right? So if it's a hard problem, right? We sell things for further manufacturing, right? I was just talking to Carter with this recently, and by creating a final product for infusion, right? That's a therapeutic product, right? And if you just sell that to somebody, they can inject it and really mess people up, right? And if you're going to make that therapeutic product, you are going to then run that clinical trial. You're gonna make sure it's used in the appropriate fashion. There's a lot of things to do, right? And that's a very different business model than what we've created. There are companies that have tried to do this, but sorry, you're gonna.
I was just going to comment on Vinay's presentation. What I want to emphasize is that the CAR T cell program was specifically designed here to address this problem, because we're addressing rare and pediatric cancers for which there isn't a commercial vendor that produces it. But we really provide that at no cost around the country to trialists who are interested in treating these rare cancers. I think that's a model where the NIH could really be helpful. And it could give you a niche that you could fill if you were able to address how to democratize.
Yeah, I mean, it just costs money, right? So, and I would say that, if FNL wants to establish a MSE process that they can make final doses, right? We can help with that for sure, right? But the funding's got to come from someplace, right? It's all the stuff's really expensive, Okay, there's a question back here as well. Yes.
Yeah, I just had like an EV question because they're super cool. Like I was wondering what methods you started to trust for characterizing the EVs and establishing efficacy for checking them.
So yeah, great question. Yeah, I mean, these nanoparticles are challenging to characterize. We do have several EV analytical methods that we have in house. We use the nanoflow cytometer. It's more identity, right? But understanding what's in these is really, really important. And then we do have some cell-based assays, right, that we use to look at the biological activity. But they're used in so many different things, and we're not a therapeutic company. So we then have to work with therapeutic companies to make sure that they are, characterizing and developing, assays for the EVs that are relevant to their therapeutic, indication, right? So
specific to what they're doing.
Yeah. And so, yeah, we have an angiogenesis assay. We have a macrophage, polarization assay, right? But unless someone is taking it into an indication where this is in wound healing assays, right? So now, I think one of the commercial applications that people are looking at is skin aging, right? Another way to look at it is beauty, right? So and you can get these exosomes are being used in cosmetics as well, right? And so and there's a lot of data out there to support that. Yeah, you throw these on 3D skin models, right? And they upregulate collagen and elastin, and get nice fuller skin, right? And these are being used for those types of applications as well. But it's really you have to know the product you're trying to develop, and since we don't develop direct to consumer products, nor will we, we just have to be judicious in which assays that we develop based off of what our customers need, right? So we do have an analytical development services part of our process development services, and we do help customers with that. But yeah, but unless you know exactly what product you're going after, it's
Thank you.
Yep.
Oh
All
my
right.
God, what?
Thanks. Thank you.
Hello, everyone. First of all, thank you for inviting me to come here and speak today. I really appreciate the opportunity. My name is Brian Paszkiet. I've been working at Miltenyi Biotech for going on over 8 years now. And my role at the company is really a technical role where I train customers and help them and show them how to use our technology in the field. I wanted to give you a little bit of a broad overview about Miltenyi, just in case some of you haven't heard of the company, as well as to talk about some of the products that we offer that enable making cell therapy products. So our mission at Miltenyi is to advance the scientific understanding and medicine by providing solutions for cell and gene therapy and biomedical research. We obviously have a very large portfolio of different tools that can be used in the cell and gene therapy field. I'm not going to go through this step by step, but I would like to start with some of our instruments that we offer. And the first one that I can't, skip, obviously, because it's been around for basically going on 25 years, is the CliniMACS Plus. So the core technology of Miltenyi is really using magnetic beads to essentially enable separating different cells out of a blood product. So essentially you can imagine, a patient needs, let's say, a CAR-T treatment. Well, you can collect an apheresis from that patient and then use, say, CD4 and CD8 beads to isolate the T cells away from all the rest of the cells. That is kind of the tenet of the core technology of Miltenyi. And so the CliniMACS Plus, has had a lot of success in making in doing what we call graft engineering. So in taking, say someone has a cancer and they undergo, chemotherapy, and now they need to recover their immune system. So they can often go to a related donor, get a mobilized leukapheresis from that, possibly family member or matched donor. And we can use the CliniMACS Plus to isolate the CD34 cells from that blood product. That then gets given back to the patient and they can reconstitute their immune system. But that's that was really where the clinical aspect of Miltenyi started, is with the CliniMACS Plus. And you can not only do the CD34 aspect of it, but you can also use it to isolate a variety of different cells as well. And I will say that this is still a very widely used instrument in the field. A lot of times people think we've moved beyond it, but it is still actually a very robust product for us. Now, I would like to move on to more complicated manufacturing scenarios. So what I just showed you was the CliniMACS Plus, which really has the focus of automating the cell enrichment or cell depletion process, meaning that the end user has to do a lot of upfront manual manipulations before you go onto that instrument. But you can imagine cell and gene therapy manufacturing is quite complex and can involve quite a number of different manual steps. That introduces risks because obviously as you go from one step to the next, possibly operating in a biological safety cabinet, there's always the possibility that you might have some issues with risk of contamination and things like that. So our goal was really to build something that would automate all the different parts of this process. And we did that in the CliniMACS Prodigy. So this is essentially a device that now automates everything from the beginning to the end of the process, starting with a leukapheresis product or a blood product, washing it, labeling it with the magnetic beads, isolating the different fractions of cells, also enabling activation, transduction, and cell culture, and then final harvest and formulation all in this one device in a completely closed fashion. So this really removes a lot of the risk associated with getting contaminations and things in the middle of doing a whole bunch of different manual steps. And what we're finding is that, it's really a great platform that can really take you from the early stage R&D and development all the way to commercial phase. And we do have customers that are, in countless numbers of different clinical trials, as well as also in commercial manufacturing. I'm not going to go through these step by step, but it's just really to point out that we have a lot of publications and we're involved in lots of different clinical trials and commercial manufacturing in the field. But I would like to go through some of the more detailed aspects of the Prodigy. So what you can see is that we have an integrated centrifugal unit, which enables cell washing as well as cell culture. We have that central tenet of our technology, which is the magnet and the column. And that's what we can use to automate the cell either enrichment or depletion process. We have the integrated tubing set, which is a closed system processing, enables attachment of different products on the top, movement throughout the tubing set, as well as collection of the final products on the bottom. Peristaltic pump drives the flow of the fluid throughout the whole system using the different pinch valves. We also have gas mixing unit, and this is what enables doing cell culture on this device. We can maintain the temperature at 37 degrees, as well as provide CO2 to the headspace of the chamber. Everything is driven through the touchscreen interface, so it really works well in clinical manufacturing, works well in clean rooms. And so basically everything that the operator has to do is simply explained on the touchscreen interface. And then finally, an integrated tube sealer and barcode reader for reading all the products and sealing off the bags at the end. We have built-in processes on this device, and the goal of Miltenyi is really to provide these tools to everyone. We don't charge individually for all the different programs. Actually, everybody that buys a Prodigy gets all of these programs. As we develop new programs, we automatically put those on customers' instruments. But we do also offer a service whereby a custom application can also be developed. So depending on, sometimes people want to do something a little bit differently than what we've envisioned, we also enable them to make custom processes on this device as well. And finally, I would like to point out that we are also going into the realm of having GMP compliant software. And so that's something that's been told that's very important to us, especially in the commercial field. Here is just a number of the different types of programs that we provide on the Prodigy. And so basically we're automating all of these different types of processes. I'm just going to highlight a few of them and show you a little bit more detail about each one. One of our most popular programs is called T-cell transduction, and this enables making CAR T-cell in a single program, in a single device. It essentially starts with isolating T-cells, activating and transducing them, and then expanding them and formulating them all inside the device. And I'd like to point out that depending on how long you grow the culture inside the Prodigy, you can achieve upwards of about 5 billion total T cells, with a total of approaching 2 billion CAR T cells, which is certainly relevant for treating patients in that CAR T field. Another highlight of what we can do is generating NK cells on the Prodigy as well. We do that essentially by depleting out CD3 cells first, and then enriching for CD56 positive that gets rid of the NK T cells. And then the Prodigy can then be used for essentially manufacturing those, say, into a CAR NK cell or other types of workflows as well. Pointing out that, final cell numbers approaching 3 billion total NK cells is possible. Obviously, these things tend to be donor dependent. And then finally, I want to point out some of the workflows that we have for working with hematopoietic stem cells. So you can use the CliniMACS Plus or the Prodigy actually to enrich CD34 cells out of a blood product. And then those isolated CD34 cells can then be engineered inside the Prodigy tubing set as well, following a case of transduction and mild expansion and final harvest. This can give you, genetically edited CD34 positive cells, showing that the initial enrichment process only takes about 1 hour of hands-on time, but gives very, very high cell viability and cell purity. And also the culture and transduction process, very minimal hands-on time. Manufacturing only takes a few days and gives you very high cell viability in the end as well. Then just to discuss very briefly kind of the vision of how this is built into larger manufacturing sites. So, we kind of advocate for ballroom type facilities, and this can be used both in centralized as well as decentralized point of care manufacturing. Basically giving you seamless scaling of your process from the small scale development part all the way up to commercial. And we are working with clients on this right now. And like I said, pointing out that, you can have the notion of having more localized point of care centers for doing the manufacturing. And again, I will say that this is being done today already. I know point of care manufacturing is a really hot topic nowadays, but essentially, if a hospital or a site has a Prodigy, they can do point of care manufacturing right at their site. So it obviously works in that type of scenario, as well as centralized manufacturing, where the blood products are already sent to a central facility, and that is also something that's being widely done today as well. Well, I thank you for your attention, and I will welcome any questions. Yes.
Thank you for your talk.
Sure.
Question on your NK expansion.
Mhm.
So 3 billion cells, is that like, you said, of course, donor to donor dependent.
Mhm.
Is that like the best you've gotten, or is that like typically like the average of like what the expansion is?
No, I
like?
think we've been able to get better than that. I think that's more like the
That's, like,
average.
your
Yes.
average?
Yeah.
Okay. And that's under activation?
Yeah.
Thank you.
Mhm. Anything else? Anybody online?
I had a question regarding the setup.
Yes.
So do you buy kits that are prescribed already for what you're trying to do, and then you have tubing kits? Like, is it plug and play so that my kid could do it, or do you still need to have an experienced operator who's setting it up, managing it?
That's a great question, honestly. I mean, I've been doing this for 8 years, and I really know that I can train somebody. I could train your kid how to do it. I know the tubing set looks very daunting and everything, but it's really engineered in a way that makes it very easy to install, and it's all guided on the screen. The other question is, we do have several different types of tubing sets that are kind of purpose built for different processes. So depending on what you want to do, that will dictate what tubing set you need to get and what products and supplies you need to get.
then
And we've worked that out.
The operation, well, it looks like you're booking the machine for up to 12, 14 days
Mhm.
per blood product. And
Yes.
The total consumable cost for that, of course, is depending on what you're doing, but
Mhm.
what's the average cost of consumables?
Oh my gosh. I mean, that's kind of a hard one to answer. I mean, I think for a typical CAR T manufacturing, let's say, $15,000 is probably around the ballpark of what it might cost to do something like that. Obviously, every process is going to have a different level of cost. And also to your point about, another criticism I hear sometimes is that, oh, I have this expensive machine and I'm keeping it occupied for such a long time. Well, you can see what people are doing in the field, and we also advocate for it, is okay, you can use the Prodigy to do complex early parts of your manufacturing process, but then have the cells brought out into another device for cell culture. And there's totally nothing wrong with that. And it is widely used in the industry as well.
Do you use the G-Rex flask?
We have, yes, absolutely. Yes. So, I mean, to be honest with you, we can, and I've worked with clients, I can't discuss the details, but I mean, I've worked with clients that we've developed custom applications that essentially do a cell processing upfront, and then it transfers the cells and all the cell culture media directly into a G-Rex. The Prodigy automates all of that. So it's very powerful. Yeah. Yes.
It sounds like you offer over-the-air updates and automatic program coming into this machine.
Mhm.
Have you had any problems with maintaining product qualification, since it's a qualified system that the software does not remain constant?
Yeah, so the way we do that, I mean, I'm not a regulatory expert. I will first say that. But what I will say is that obviously, just like everyone else in this field, we have all the drug master files and all that for the device. What we do when we develop new programs is we basically add that to our filing with the FDA. So it still maintains its regulatory status, and we're obviously, we have change control and everything. So we're showing that we haven't made any changes to other programs when we add this new one, and that type of thing. So that's how we've maintained control of that. Yeah. All right, thank you.
All right, so let's have another round of applause to our amazing speakers. Great. I would also like to get an opportunity and thank our co-organizers, which is the Frederick County Chamber of Commerce, and the sponsor for today's event, MSIC Company. So thank you so much for that, for the coffee. Also, our next event will be focused on the AI-powered and computer-driven drug development. So it's going to be February 19th. If anyone is interested, please come and join us for that. And when you leave, please take a look at the exhibit of pieces of art that we have here in the atrium. It was provided by the Frederick Arts Council. They invited the artists to exhibit something, and they were inspired by the science, including what we are doing here at the Frederick National Lab. So please take a moment and look at that. If you want to look at more pieces, they are available in Y Art Center in downtown Frederick. So grab some coffee, bagels, if there is anything left. And thank you so much for coming. And thank you everyone online as well for attending. Thank you.
Thank you.